Today we're starting with Unit 7, where we're going to discuss genes and inheritance. So family resemblances. Your mother and your father are both going to contribute equally to your genetic makeup. Genes are going to be the reason why people are going to resemble others in their family, such as their mother and their father. So offspring often resemble their parents because of these instructions. They can be instructions for physical appearance, which we usually see. It determines the hair color, the eye color, the color of the skin, the height of the person. It could be biochemical instructions, which are going to tell the body what to do and when to do it, when to grow, when to go into puberty. Other biochemical things are just simply going to be enzymes that break down specific things that you take into your body. Also, it helps with behavioral traits. And so it could code for if you prefer certain foods or not, because that's going to be determinant upon what genes that you have. So with each parent, they're going to have 2 chromosomes for every chromosome there is. So chromosomes 1 through 22 are referred to as your autosomes, and then chromosome 23 is going to be the sex chromosomes. On those chromosomes, you're going to find specific places where genes are going to be located. Looking here, we're looking at the maternal chromosome pair. That's going to be the darker teal with the orange stripes on top. The chromosome location, or if you remember from previous chapters, the loci or locus, is going to be at the top of this chromosome. Looking over at the father, who's going to be a lighter colored teal, you see that same gene that we are looking for. One of those chromosomes is going to make it into the egg. One of the chromosomes from the father is going to make it into the sperm. When it goes through meiosis, it's going to divide these up and put one of each set into the egg or the sperm. During fertilization, the egg and the sperm are going to come together, and the gametes are going to merge so that child has one from each parent. And so you see below, you have the darker teal from mother and the lighter teal from the father. And so whatever specific trait that child inherits, it's going to be on that locus of the chromosome, and it will go together sometimes depending on what genes, which we'll talk about later. It might have one that's going to be dominant over the other. They may blend, or they may both show themselves. So how can a single bad gene make you smell like rotten fish? Well, there's going to be a gene for an enzyme called FMO3, or flavin-containing monooxygenase 3. This is going to be caused by a defect in gene on chromosome 1. And so the mother, of course, has 2 chromosome 1s. Father has 2 chromosome 1s, so they can both be carriers of this gene, but not realize they have the gene because it's going to be a dominant and recessive trait. Or they may not carry the trait at all, or one may carry the trait, or both may have the trait. It just depends. So what this enzyme does is it's going to break down a chemical in the body that smells like rotting fish. This smell is going to come out in the urine, the sweat, the breath as it's excreted when someone's talking. And so this is going to cause that person to smell like rotten fish. The smell is actually going to come from within the body, so it's going to be hard to keep that smell down. You can brush your teeth, wash your mouth out with mouthwash, but over time, once that mini smell starts to diminish, it will start to come out in the breath again. You can take a shower and wash with soap, put on some perfume, but over the period of the day, you do release sweat and urea and things throughout your pores, so it will start to come out. It is referred to as an autosomal recessive trait. Autosome because it's on chromosome 1. Recessive because if you have a dominant trait along with it and it's heterozygous, then you will not see this trait. And so it can be passed on to the offspring. Now looking at the 2 parents here, you see the father has a dominant regular trait and he has a recessive FMO3 trait. So does the mother. She has the dominant regular smell trait and the recessive FMO3. This is the possibility of their offspring when you work a Punnett square. And so I will go ahead and let's just make a little spot right here so you can see, and we'll work this Punnett square. Let's put dad up top. Now it doesn't matter if dad's on top or on the side, or mom's on top and the side. You're still going to get the same outcome. And so here's the Punnett square. And so you bring one over, bring one down. Now bring that capital over again, the lowercase down, bring the lowercase over, and always try to keep the lowercase behind the uppercase. If there's an uppercase, try to keep that in the front. And then one down, one over. And this is your possibilities. And that's what you see in these pictures below. And so you end up with one child who is not going to carry the gene. It's going to have 2 normal traits, so that's 25% out of this category. You're going to end up with these 2 right here that will carry the trait, but you're not going to see it. So they're just going to be referred to as carriers. That's 50%. And then you're going to have a possibility of one child actually receiving both of those recessive traits. And so they're going to be homozygous recessive, but they will have the FMO3 trait. And so here, looking at this, they put it in another picture form so you could look at it. The normal version is going to be the straight line going across the chromosome, and notice it switched the location. So let's say this is chromosome 1, and the locus is going to be underneath where it indents, so that's where you're going to see it. The trait for the FMO3 gene is going to be the zigzag. And so let's say that the egg ended up with the FMO3 trait, the sperm ended up with the FMO3 trait. When it came together during fertilization, you do produce a child that is going to be homozygous recessive and show that trait. Looking at this, how could 2 normal parents have a child with Fischer syndrome? I'll give you a moment. All right, the answer is number 4. Both parents had 1 normal and 1 mutant allele for the FMO3 trait. So some traits are going to be controlled by a single gene. Heredity is going to be the passing of characteristics from one parent to offspring through their genes. And so each parent passes these genes on, and sometimes they, like I said, combine together. Sometimes one's going to be dominant over the other, and then sometimes both of those traits are going to show. Some examples of single gene traits that you need to remember are going to be things such as a cleft chin. Now the cleft chin is going to be dominant. The non-cleft chin is going to be recessive. Unattached earlobes versus attached. The unattached are going to be dominant over the attached, which are recessive. And then the widow's peak. Widow's peak is going to be dominant, and the straight hairline is going to be recessive. Now, all of these are going to be controlled by one gene that's going to be found at a specific loci or location on a chromosome. Over time, we've used things called selective breeding in organisms to get specific desirable traits that we want to see, whether it be an animal or a plant or something that we are trying to produce. So what's popular now are going to be things such as these teacup puppies or dogs. There's also going to be these mini ponies. Sometimes you see the small miniature goats running around. So these are going to be things that people actually go in and they selectively breed for. And so they find a trait of the smallest, let's say we're going for a dog. And so with that trait, you find both parents are going to be the smallest that they can find of those litters. They will bring them together, let them breed, and then they'll take the smallest ones again from separate litters, put them together, and continue to breed it like this until they get very small animals. They can also make them grow bigger and breed for those traits for them to be bigger, or breed for traits for them to be hardier or sturdier. If it's a crop, they can breed for ones that have the biggest output, or maybe the ones that survive drought the most. So single gene traits, some traits are going to be determined by instructions that an organism carries in one gene. Lots of traits that we have actually are going to be a combination of several different genes that come together. For example, skin color and height will be made up of 6 separate genes. So you inherit 3 from mom and 3 from dad. They come together, they combine together, and that's what gives you a mixture of your height or your skin color. So some examples of other single gene traits is going to be fur length in cats and coat color in cats. So some traits, like we said, are going to be determined by instructions that we carry on a single gene, and this is going to be a more straightforward pattern of inheritance. For example, when we talk about diseases, Huntington's disease is going to be a single gene trait that can be inherited. That is going to be a progressive degenerative breakdown of nerve cells in the brain. So unfortunately, these people, once they're diagnosed, do not live incredibly long. Usually it's going to be around 3 to 5 years after diagnosis. Okay, there's going to be multiple gene traits too. Like we said with, let's say, skin color and height. Now they're even thinking that eye color is going to be multiple genes that come together. So most human characteristics, like I said, are going to be multiple gene traits. So first, we're going to examine the mechanism by which a single gene trait passes from parent to child. So Mendel is going to be our scientist who's going to actually delve into this and look into this very closely. And he's going to conduct many experiments in his lifetime. So starting out, inheritance, or the child, actually was thought to have been created and carried forth to the womb from the father. It was thought that every sperm had a completely formed, minute, tiny little human being inside, and that the father deposited that child into the mother's womb, and then the mother was responsible for growing that child. Now, anything that she might have done during her pregnancy, if she stayed upset, or if she ate things she wasn't supposed to, or did things she wasn't supposed to, that would be what caused harm to the child, or that would be what interfered with that child's growth. So the father, he deposited a perfectly developed human being, and then it was given off to the mother, and the mother was the reason why something might have happened after that deposit. Now, this little child inside of the sperm was referred to as a homunculus. And this theory was believed starting around the 1600s and actually carried into the 1800s. So Gregor Mendel, he was born in 1822 and lived till 1884. He's going to be a scientist and an Augustinian monk or friar, and he's going to work with 7 characteristics in a plant that he grew anyway in the monastery. He's going to work with a pea plant. And so some of the things that he's going to look at with those pea plants is going to be plant height, pod shape, color, seed shape and seed color, and flower position and color. These are all going to be characteristics that he's able to see and observe quite easily, and then can use those traits when he's fertilizing and cross-fertilizing to see if they're carried on from parent to offspring. And so he works with 3 key plant features, and these are going to help make his experiment work well. Flower color, which is going to be easy to see, pea color, and pea shape. 3 research features is that he chose a very good organism. It's something that does not take very long to grow, and it's easily observable and manipulated. He's going to choose features that are easily visible and repeated crosses between similar plants first, and then he's going to cross those that are not similar. Segregation. You've got 2 copies of each gene, but only 1 copy into each sperm and egg. So, 3 ideas that Mendel used for explaining this pattern of inheritance is that each parent puts into every sperm or egg it makes a single set of instructions for building a trait. These instructions are going to be called a gene. Offspring are going to receive 2 copies of the instructions for any trait, and each of these copies is called an allele. So, for example, if you're talking eye color, you can have an allele for brown eyes and an allele for blue eyes. And so how we would write that, let's just use B's. Brown eyes dominant, so I'm going to use a capital letter. Blue eyes recessive, so I'm going to use lowercase. Recessive's always lowercase, dominant's always capital. So each one of these that you see is going to be an allele, or instructions for a trait. When you put them together, you create the genotype, and that would be big B, little b. And third, the trait observed in the individual depends on 2 copies of the gene that it inherits from its parents. So it could be homozygous or heterozygous. Now, homozygous would be two of the same trait, and so I could have two dominant alleles for brown eyes or two recessive alleles for blue eyes. Both of those are the same letters or the same capital or lowercase, so they're considered to be homozygous. Or, like what I just showed you before, they could have brown eyes but have a trait for brown and blue, and when they have one of each trait, it's considered to be heterozygous. So looking at these alleles, we talked about dominant and we've talked about recessive. So dominant traits are going to be those that are seen present with a recessive trait. So that means you may have the recessive trait, but the recessive trait is not seen. So the one that you do see is going to be referred to as dominant. And like we said, it's going to be represented by an uppercase letter. The recessive traits are going to be the ones that you only see if you have both recessive traits. So if you're carrying a recessive trait but do not see it, then that one will be masked. And these are going to be represented by a lowercase letter. Now looking below, you see the Punnett squares of how they're put together. So you can have two parents with brown eyes but carry a blue-eyed trait, and you didn't realize you had a blue-eyed trait, let's say. So if you and your partner were crossed and had possibilities of children, there's going to be a possibility that the child's going to inherit both dominant brown-eyed traits. That's 25%. There's going to be a possibility that they're going to be heterozygous like both parents, and that's going to be a 50% possibility. And there's going to be a 25% possibility that the child will come out with blue eyes, even though both parents have brown. With this, what you see is going to be referred to as the phenotype, and what the genes are made up of is the genotype. And so here you see that you're going to have 1, 2, 3 that are going to show brown eyes. So the phenotype here is 75% brown eyes. You're only going to have one that shows blue eyes. And so the 25% chance that child will have blue eyes. Dominant traits are going to mask the effect of recessive traits. And so when you look at these pea plants above, these are going to be like the ones that Mendel used. He's going to cross true breeding purple flowers with plants that are going to be true breeding white. And so when he crosses these, the purple are going to be dominant, and he's going to end up with purple flowers. If he takes two of those flowers that he crossed from the original, now remember, they do have a white trait because they had a white parent, and they do have a purple trait because they had a purple parent. When you cross these, only then will you see some of these white traits start to reappear again. Before, that dominant took over, and all you saw was purple. So this is referred to as Mendel's law of segregation. According to Mendel's law of segregation, only one of the two alleles for a gene is going to be put into a gamete. At fertilization, offspring receive from each parent one allele for each gene. And so we take two heterozygous parents. That means they each have different genes, one white, one purple. You assort them, separate them into sperm or egg, and then you put them together through fertilization. And then that's how you determine which type of plant you have once it grows. So observing an individual's phenotype is not going to be sufficient enough for determining its genotype. Remember, phenotype, think of photo. That's what you see. So as far as the brown eyes and blue eyes were concerned, if you have a trait for both brown eyes and blue eyes, remember, you're just going to see brown eyes. You cannot see the masked blue-eyed trait. So how do we analyze and predict the outcome of crosses? We're going to assign symbols to represent the different variants of a gene. Generally, like I said, we're going to use uppercase letters for dominant alleles and lowercase letters for recessive alleles. The outward appearance of an individual is called the phenotype. Remember, that's what you see. The underlying phenotype is the genotype. And so that's going to be the actual alleles that it's made up of. And so this is the genetic composition. This is what the letters are, capitals and lowercases, heterozygous or homozygous. That's going to be the letters it's made up of. Remember, phenotype's just what it is showing. So phenotype, little or no pigment in the eyes, hair, and skin. Genotype is homozygous for the recessive allele for albinism. And that's what we see here in this deer. So homozygous genotypes are going to have two of the same allele for a gene. So it could be two dominant alleles. Two capital D's. Two recessive alleles. Let's use two lowercase d's. And these are often referred to as true breeding. Now with true breeding, that means that their genes are pure. It's just of one type. It's not going to be a hybrid or a mix. And so you see down below, they give you AA or little a. Now with a hybrid, that's where you're going to have a mix of genes. So you can have a capital and a lowercase together. And so you're going to have a dominant and recessive. And like we said, most of the time you see that dominant over the recessive when you're talking about one gene traits. And these are often referred to as hybrids. So purebred or true breeding, if they're homozygous, homo means of one type, hetero means of other types. And so the heterozygous are going to be hybrid. So how do we analyze and predict the outcome of crosses? Well, first we assign those symbols to represent different variants of a gene. And generally, we're going to use an uppercase letter and a lowercase to represent these in what we call a Punnett square. And so with the Punnett square, it's going to be a diagram that's used to predict an outcome of a particular cross when you're breeding. For a particular trait, an individual may carry a recessive allele whose phenotypic effect is masked by the presence of the dominant allele. And so here we're showing the green and the yellow peas. So we have a homozygous recessive green up top, and we're going to cross it with a heterozygous yellow on the side. Now it's going to have one of each gene, but all you see is yellow because that yellow is going to be dominant. And so when you cross those, you're going to bring one over, bring one down, bring one over, bring one down. Let me show you that. So bring one over, and you bring this one down. And then for the next one, you bring it over, and you bring this one down. And for the bottom, bring it over, and bring this one down. And for the last one, you bring one over and bring one down. And so that's how you figure it out. So with genotype, you're going to have two possibilities. You have the heterozygous possibility, which is 50%, and you have the homozygous possibility, which is 50%. So it's a 50-50 depending on what color it's going to turn out to be. Now, if you're talking about phenotype, what color, in this case, is also going to be 50%. This one will be 50% yellow, and then this one's going to be 50% green. So in this case, it worked out that they're both the same. Genotypes versus phenotypes. Here's a Punnett square for albinism. So we take a mother who's going to be albino, and then we take a father who's going to be dominant for normal pigment. You cross those two, and you end up with a 100% chance that you're going to come out with a normal pigmented giraffe. All of them will be heterozygous, but they do have normal pigment. Now, for cross 2, you can take a mother who is heterozygous, carries a normal pigment, and then an albino pigment. Then you have the father who carries a normal pigment and then albinism. And you can cross them and you see what you end up with. There's going to be a 25% chance, or 1/4, that it's going to be dominant for normal pigment. 50% chance it's heterozygous and will carry the albinism gene but be of normal pigment. And a 25% chance that it's homozygous recessive and it will have albinism. So probability and chance are going to play central roles in genetics. Every time you go to have a child, or every time that you have offspring, whether it be for a plant or an animal, it's always going to fall the same way. Just because, let's say, let's go back, just because you ended up with a giraffe here that was albino doesn't mean that this one gets crossed off the list and the next time you have a giraffe, it'll have to be one of these three. No, every time it starts over with the same 4 possibilities. So chance is going to be important in genetics. So probability does have a central role in genetics for 2 reasons. The first reason is a consequence of segregation. Each gamete is going to receive only one of 2 alleles, and it's going to be impossible to know which of the gametes will receive this, which ones get which allele. After that, we talked about in the previous chapter crossing over of those sister chromatids. And so even though one might receive one trait, they can still swap during prophase 1 of meiosis before they get pulled apart completely and segregate. The second reason is that fertilization, too, is going to be a chance event. You don't know which sperm or which egg is going to be fertilized. So any gamete produced by an individual heterozygous for a trait is going to have a 50% possibility of carrying the dominant allele and a 50% possibility of carrying the recessive allele. If the individual is homozygous for the trait, then they're going to have 100% of their gametes carrying that allele. If male is going to be heterozygous for albinism and the female is going to be homozygous for albinism, what's going to be the probability that their child would be homozygous for albinism? And so what you're going to do is you're going to set it up and do a Punnett square. And so let's put the dad up top. He's going to be heterozygous, a dominant and recessive. Mom on the side, that's 2 lowercase m's. So remember, always try to keep your capital in the front. So bring the big one down, little one over, little one down, little one over, big one down, little one over, little one down, and little one over. And so that's going to be your possibility. You'll end up with a 50% chance that it's going to be homozygous recessive and that offspring will be albino. And you'll end up with a 50% chance that it's going to be heterozygous and you're going to just be a carrier for albinism and it's not going to show that trait. It will have normal coloring. And so here, they actually give you a picture and show you what they're doing. Same thing with Tay-Sachs. They put it together and cross it. So a test cross is going to enable us to figure out which alleles an individual carries if we don't know. So we can test cross white alligators. And so if you'd like to produce white alligators through a mating program, because they do draw more people into an exhibit or an attraction or to like a zoo, then you can take 2 alligators that you're not certain of their genotype and you can do cross test with them. And so they might be homozygous dominant or they could be heterozygous. And so either case, their phenotype is going to be normal coloration. And so you could take a alligator from, let's say, ones that you already have if you have an albino alligator, or you could borrow one from another facility and you could do a cross with them. And so if you cross it with a normal colored alligator, then you can come out with the possibility. So if the father was normal color but had both dominant genes, then this is going to be your possibility, and you know that both of those now, or all of those now, will carry the recessive albino gene. Or if the father is a carrier, when you cross them, this is what you're going to end up with. And so you'll end up with 50% normal coloration that carry the albino gene, and you'll end up with 50% albino offspring. So how to determine genotype? In a test cross, that individual that exhibits a dominant trait but has an unknown genotype is going to be mated with the individual that's homozygous recessive. So the phenotypes of the offspring are going to reveal whether the unknown genotype individual is homozygous dominant, all of the offspring are going to exhibit that dominant trait, or if they're heterozygous. And that's where half the offspring should show the dominant trait and half show the recessive trait. So using pedigrees to decipher and to predict the inheritance patterns of genes is going to be used when you're trying to go through, let's say, the family tree and see where a trait came from, or you're trying to determine if the offspring might contain a certain trait. And so a pedigree is going to be simply a type of a family tree. So to create your family pedigree, information is going to be gathered from as many family members as possible. Starting from the bottom, each row in the chart is going to represent a generation. And so looking over here at the pedigree that we see that we have different generations. We start from the bottom and work our way up. Now the top one that we end with is going to be called the first generation, even though we started from the bottom. Then the one below is going to be your second generation, then your third, and then your fourth. So we go down 4 generations here. You're going to list all of the children in their order of birth and whether or not they exhibit a particular trait. Now in this case, if they have a trait, then we're going to color that box in. So you see that only some of these children are going to exhibit the trait hemophilia that we're looking at in the royal family. Then we're going to work our way up the pedigree. The parents are going to be indicated, and above them, their parents, which would be the grandparents, and you're going to go as far back as data is going to be available. And so this, we're looking at Queen Victoria and Albert. And so you see their kids. They had, first thing you have to know is male and female. So male is going to be a square, female is going to be a circle. And so they're going to have 1, 2, sorry, it should be 2 and 3. They have 3 boys, and you see the girls. They have 1, 2, and 3 girls. And so looking at those, only one of their children is going to inherit that hemophilia trait. And so that means that they don't clot properly when they're bleeding. And so they tend to be free bleeders. Going through, because Albert and Victoria did not show that trait, we know that it's going to be a homozygous recessive trait for it to show itself. So both of them had a normal clotting trait, so it covered up their hemophilia trait. And so they're just simply carriers of this disorder. Coming on down, you see that Alice had 3 children. She had 2 girls and a boy. The boy is going to inherit that trait. Looking over at the other, you see that Beatrice is going to have 3 children, and the 2 boys inherit the trait as well. Going down to the 4th generation, you see that one of the daughters has 3 boys. 2 of the boys inherit the trait, 1 does not. Coming on over, you see that the other daughter has 2 children, a girl and a boy here, and you see that boy inherits the trait. Leopold had a girl. She did not exhibit the trait, but the boy did. And then you see the other girl here that Beatrice had. She's going to have 4 children, and the boys inherit that trait. Now, looking at this, notice how the females are never showing the trait. They're simply carriers. This kind of alerts us and lets us know that this is going to be a sex-linked trait. So that means it's going to be carried on the X chromosome and inherited from the mother. Since boys only get an X chromosome from their mother if they get the faulty copy or the copy that is coding for hemophilia, then that's what they exhibit. They have no other alternative. But since the mother gets a second X from the father, then she has a chance of having that normal clotting trait, and so she doesn't exhibit it even though she carries the gene. So she's likely to pass it on to her sons, and she can even pass it on to her daughter as a recessive trait, but chances are that daughter might get a normal clotting factor from her father. So squares, like we said, represent males. Circles represent females. The shaded area is going to indicate that trait of interest, so those with hemophilia. And if the individual exhibits that trait and neither parent have it, then you know it's a recessive trait. So analyzing which individuals manifest the trait and which do not. So the pedigree is going to be a useful tool that helps you do this. So you see two sets of grandparents here. One's going to exhibit trait of interest, and that's going to be that grandfather on the left-hand side. And so you see that they have 3 children. 2 of those 3 children, a girl and a boy, are going to inherit that recessive trait. You know it's recessive because they have the second boy in the middle who doesn't get the trait, but he has genes from his father. And so sex-linked traits are going to be traits that are carried on the sex chromosomes, which is going to be the X chromosome. Since males only have one, like I discussed before, they're going to be more likely to exhibit this trait. So males get one X chromosome and it comes from the mom. That egg contains the X chromosome. Females receive 2 X chromosomes, one from mom in the egg and then one from dad, which will be in the sperm. And so if you see it, let's go back here real quick. So the father here, since he only has one X, then he does exhibit the trait that we're looking at. And this is red-green blindness. Mother, we don't see any traits. That means she has both normal vision. But when they have children, since the dad can give one X to the little girls, then you see those girls getting that colorblind trait. And the only X he had was colorblind. Mother gave them a non-colorblind trait, so they are going to be carriers of this disorder, but they don't exhibit the trait. Another sex-linked trait is going to be found in dogs. It's called anuria. This is going to be a condition in dogs and in other animals actually, where they're born without a tail. They kind of just have a little nub there. And it's going to be an inherited recessive trait that comes to them. So traits are going to be a mode of inheritance that's not always going to be completely obvious. Some traits may not show complete dominance, and many traits are going to be influenced by the environment. Now, let's come back here. So if we're going to do sex-linked traits, let's go ahead and do a Punnett square. So give me a second. And this is going to be important that you know how to do sex-linked traits. So let's say that we have mom, and so girls or females are going to be XX, males are going to be XY. So since it's a sex-linked trait, it will be carried on the X chromosome. So the Y does not have the trait. So how you're going to do this, let's talk about the anuria. That's going to be a recessive trait. And so let's say the dad had anuria. That's going to be where he has a little nub for a tail. He's born without a tail. And let's say mom is going to be heterozygous for the trait. So capital means she has a normal tail, but she carries the trait for no tail. And so now we do the cross. So we have to put XX because you brought one over, one down, then XX again, because when you have children, it's 50-50. So that's your 50-50 up top. Down below, you have XY and XY again. Now, you're going to add in the traits. So dad can only give a lowercase, and remember, keep a lowercase over to the side. Mom, in this case, up above, is going to have the capital A. So this is going to make a carrier. Bringing over, we have a lowercase from dad, a lowercase from mom. Okay, you just look up top here, and it's going to come down. So all you have is a capital up there, and you look up top, bring it down, all you have is a lowercase. So in the case of a girl, you're going to have a 50% chance, if it's a girl, that it's going to be a carrier of the trait, and a 50% chance if it's a girl, it's going to have that trait for anuria, no tail. If you're looking at the boy, you have a 50% chance that it's going to have a tail, and you have a 50% chance that it is not going to have a tail. If you were talking about all sexes together, you would have 50% chance still that they have a tail, whether it be girl or boy, and a 50% chance that they not have a tail, whether it be a girl or a boy. So that's how you're going to do sex-linked traits. You have to use the X's and the Y's and then put the letters in. So why do you look at pedigrees, or why would you pay attention to pedigrees? Well, they're going to help scientists and doctors and animal and plant breeders and prospective parents determine certain things. They're going to be able to determine the genes that the individual carries and the likelihood that an offspring of two individuals will be exhibiting that given trait. Right. So how are genotypes translated into phenotypes? Remember, genotypes are the genes, that's what you're made of. Phenotypes is what you see. Sometimes we have what's referred to as incomplete dominance and codominance. And with these, the effects of both alleles in a genotype can show up in the phenotype and what you see. So here, with incomplete dominance, in which the heterozygote appears to be an intermediate between the two. So you can have a red father. Oops. Let's go back. Let me wait till this kicks in. So here you have a red father, and he's going to be dominant for red. You have a white mother. It's going to be dominant for white. And when you put them together, you see the white and red are going to mix, and you end up with a pink color. And so that's going to be an incomplete dominance. They're going to come together and they're going to blend. Now, if you take two of the pink flowers and you cross them, then you'll be able to get the white, again, 25% chance white, 50% chance pink, and then 25% chance red. Codominance is going to be where the heterozygote displays characteristics of both homozygotes. And so we see just in this picture with codominance, the father had stripes that are horizontal, the mother had longitudinal stripes. You put it together and you have the child with checks. One codominant thing that we see a lot is going to be sickle cell disease. And so with sickle cell disease, that's where you see both normal red blood cell type and you see the sickle shape red blood cell type. And so if a person is a carrier, they do not have full-blown sickle cell. Instead, they'll have some sickle cells and they'll have some regular. That's actually going to be a mutation that helps out if you live in a warm area closely found near the equator and you're likely to be bitten by a mosquito. This is actually going to keep you from getting malaria. So you cannot contract malaria if you're a carrier of sickle cell. However, if the individual does not have sickle cell disease, that means that they have both of the dominant traits to make the regular red blood cell, and so they can get malaria and become very ill, but they don't have the effects of sickle cell. If they are homozygous for sickle cell, that means they're going to have two copies of the defective hemoglobin allele, and their cells are going to be sickle-shaped. Now, the problem with this is that they're going to end up with what's known as sickle cell anemia. They're not going to be able to carry oxygen properly through the body and be able to deliver that oxygen to major organs and perform functions and things that are needed. Incomplete dominance versus codominance. So with an incomplete dominance, basically what they're going to do is it's going to blend the genes. For example, we looked at the plants. Red and white make pink. Or you could have hair color. You can take straight hair and you can take curly hair, put them together, blend them, and you end up with wavy hair. Versus a codominance where you see both traits exhibited. For example, horizontal stripes and longitudinal stripes making checks. Or here we take a red fish and a blue fish and you end up with a multicolored red and blue fish. Or you could have sickle cell anemia for the codominance. What's your blood type? Well, some genes may have more than two alleles. This is going to be referred to as multiple allelism, and that's where a single gene has more than two alleles. Each individual is still going to carry only two alleles for that trait, but it's going to be pulled from more than one trait. You get one from mom and one from dad, as usual. Our blood groups that we can inherit is going to be A, B, or O. So we refer to as the ABO blood group. The A and the B alleles are going to be completely dominant to the O. The O is going to be recessive. So if you have an A and a B, then they're going to work together and become co-dominant to each other. But if you have an A and an O, you're just going to see A, or if you have a B and an O, you're just going to see B. So the O is completely recessive. The A and B will be dominant or co-dominant if put together. So this allows you to be one of these 4 different blood groups. You could be a type A, a B, an AB, or a type O. So multiple allelism is going to occur when there's going to be 3 or more alleles for a gene within a population. An individual is still going to inherit only 2 of all the possible genes that's out there from each parent. And this shows you how you can put those different ones together. So if you are type A, you're going to show the, I didn't wait till it kicked in. Hold on. So if you're type A, you're going to have alleles for A, it's not writing properly, or A and O. Now, O is going to be represented with a lowercase I. If you're type B, your genotype can be the big B, or it could be a B and a lowercase I. If you're type AB, you're going to have that A and B. And if you're type O, you have 2 lowercase i's, and that's going to be how you represent the genotypes here. All right, so what you're basically talking about with A or AB blood or type O blood is going to be the proteins that are going to be found on the outside of the cell membrane. So let's say we have type A blood. It's going to have a specific protein that's going to be found on the outside of the cell membrane, and in this case, they're using the yellow circle there. And what that means is if you have antibodies for A, it's not going to fit on there, but antibody B will. So your body is going to react to any type of B blood if it's put into an A. Now, if you have type B blood, it's going to have a specific protein on the outside. Notice it's a different color. That's what they're using to represent it. And so that means that the immune system molecules that will attack this blood type are going to be antibody A. Now, if you have type A and B blood, you're going to have both of these proteins on the outside of the cell membrane. And so neither A or B antibodies are going to be present. And then if you have type O, that means that you're kind of like a clean slate. There's not going to be any proteins for A or B on the outside of that cell membrane. And so type A and type B antibodies will be present. And so if you put A blood or B blood basically in a type O, that means that these are going to go after it. They're going to stick to it. It causes the blood to coagulate and clump up, and it can't flow properly or carry oxygen. Just like with type B, if you put it into somebody with type, it's going to have those A antibodies. So if you put type A blood in there, these antibodies will attack it and cause coagulation. If you had type A blood here, then you have B antibodies. So if you put type B blood in, the B antibodies will coagulate. And so here kind of simplifies or shows you what blood type you might be and what types of blood that you could receive or that you can donate to. So if you're type A and you have the yellow proteins on the outside, you can donate to type A's or type AB's. If you are sick and or for some reason you need blood, you can receive from a type A or a type O. With type B blood, same thing, you can donate to type B or AB, but you can only receive from type B or type O. If you're AB, then you can donate only to AB, but you can receive from anyone because you have no antibodies that's going to attack that blood. If you're type O, then you can donate to anyone, but you can only receive type O blood because you have both A and B antibodies, so it would coagulate with A, B, or AB blood types. Multigene traits. So how are continuously varying traits such as height influenced by genes? Well, these are referred to as polygenic traits, poly meaning many. So a trait that's going to be influenced by many different genes. Unlike plants, where two alleles determine height, in humans and many animals, there's going to be many genes that can influence height. Also, since many genes can influence, they can stack on top of each other and be called an additive effect. So what happens when the effects of alleles from multiple genes all contribute to an ultimate phenotype? Well, with these additive effects, it's going to describe what happens when the effects of all alleles from multiple genes contribute to that ultimate phenotype. You can have this with skin color, so it can be an ultimate if you receive all light color, be a very light colored skin, or if you happen to receive all darker colors, be a darker colored skin. Same thing with height. You can be extremely short or extremely tall, or even eye color. So one of the genes is going to be on the 15th chromosome, and it codes for an enzyme that converts testosterone to estrogen. This enzyme is going to influence height because estrogen helps bones to fuse at the ends and stop growing. So recent research is going to identify two genes that help determine height in humans. Individuals with the tall allele for both genes are going to be taller than those with one tall gene. And individuals with one tall allele, in turn, are taller than people that don't have the tall allele for either gene. So when you have both of the alleles, it's going to be called the ultimate phenotype. With eye color, it's going to long believed to be controlled by a single gene with a dominant brown eye allele and a recessive blue eye allele. But now it appears that the results of the interaction of at least two genes and possibly more, a situation that's going to make more sense given a significant continuous variation in eye color, is actually going to be seen among adults. So why might computer nerds be more likely to have an autistic children? Well, that goes back to what we know as the geek theory. Interestingly, the genes responsible for autism may also influence desirable characteristics. Behaviors that cause problems when manifested in the extreme maybe turn out to be beneficial when they are less extreme. And so we see things such as unusual abilities of perception, being able to put things together just by looking at them or by manipulating them, analytical skills, or even extreme focus. So this idea is going to be called geek theory of autism. So if you take two parents who are extremely analytical, extremely focused, and you put them together, they're extremely smart, then they are more likely to create the ultimate phenotype and have a child who excels in specific things but is considered to be autistic. Autism is going to be the result of an alteration in numerous genes, 10 to 20 genes to be exact. So it's going to be hard to study because symptoms can vary widely. So that's why we call it the autism spectrum. It may be because different combinations of these numerous genes involved can cause different variations of the disorder, much as different alleles at several height genes may work together to produce a variety of heights. So here's the one I was talking about with skin color earlier. We're going to take 6 traits that you get from mom, and you get 6 traits from dad. Well, of course, they're going to segregate, and you only get half of those traits because they got half from their mom, half from their dad. And so here's the different ways that those eggs and the sperm can segregate and form different colorations when put together for offspring. Same thing with eye color. It's going to be a polygenic trait now, though often we use it when we're first starting out with Punnett squares as a single gene trait. So eye color was once, like we said, thought to be controlled by one gene, but now we think it's controlled by at least 2. And so know how to do Punnett squares where you can put 2 traits together. Sometimes genes are going to influence multiple traits. This is called or referred to as pleiotropy. So in pleiotropy, one gene is going to influence multiple unrelated traits. So most, if not all genes we think may be pleiotropic. So the benefit of almost having sickle cell, we've discussed earlier. So if you're homozygous and you are going to be dominant for normal blood cell type, then you're susceptible to malaria. But if you're a heterozygote, you're going to be immune to malaria. You may have some slight problems by having some sickle cells, but you have a normal cell gene as well, so you do carry blood or oxygen in your blood to body parts that need it. Now, if you inherit both of the recessive and you're homozygote for sickle cell, then you do have sickle cell disease. But you're also still going to be immune to malaria. Another gene is called the SRY gene. This is a sex-determining region on the Y chromosome. Now, knowing that only males get the Y chromosome, these are going to be what help cause fetal gonads to develop as testes shortly after fertilization. Following the gonad secretion of testosterone, other developmental changes also will occur. And so it's going to be responsible for numerous behavioral characteristics, anywhere from being more muscular to growing taller to developing a deeper voice. Why are more men than women colorblind? Well, that's going to be a sex-linked trait. And so that's going to be found on the X chromosome like we saw in the Punnett square earlier. If a man's colorblind, did he inherit this condition from his mother, his father, or both parents? Now, we talked about the X chromosome carrying this trait. That's what makes it a sex-linked trait. The egg has to be an X, and so in order to be a male, you have an X and a Y chromosome together, so that means that the egg was the one that had that trait. So he inherited it from his mother. So we said traits of colorblindness fall on the X chromosome. Here, genes produce instructions for light-sensitive proteins. These pigments make it possible to see colors such as red and green. If the person does not have functioning proteins, then they are colorblind. Females have 2 copies of this gene, so they're less likely to receive 2 of those recessive traits. And so looking here, you see the different alleles. It can be found only on the X chromosome for a sex-linked trait. So it's less likely for a female to inherit the colorblind trait or a sex-linked trait than it is for a male. Environmental effects. Identical twins are not exactly going to be identical. And so sometimes with this, environmental traits are going to kick in. And so even though they may have some of the same genes, depending on what environment they're in, depending on their stress level, some of those genes may not exhibit themselves in one twin where it would in the other. One trait that you can inherit could be phenolphthalein. This is going to be a condition known as phenylketonuria, or PKU, and this is going to be found on warning labels of things such as diet drinks or anything that contains artificial sweeteners such as aspartame. This is going to be dangerous to people who have the gene that can't process this sugar because they don't have that enzyme to break it down, so it builds up in the body and it becomes toxic. So drinking diet soda can be deadly if you carry a single bad gene. So with that, some people carry 2 copies of that mutant gene that's supposed to convert that phenolphthalein into tyrosine. Both are going to be types of amino acids. And so that mutant gene produces a malfunctioning enzyme. What happens? It starts to build up in the body, becomes toxic, it poisons the brain, leads to mental retardation, and eventually causes death if they continue to ingest this substance. So, by limiting consumption, you avoid that toxic buildup and you avoid illness or even death. So, could you create a temporarily spotted Siamese cat with an ice pick? And why? Well, genes in the environment are going to be the reason. Just because you have a specific gene, like we said, does not mean that you're going to exhibit that trait. That environment has to act on you in order for you to exhibit it. So here with the Siamese cats, you have, or your Himalayan rabbits, they are going to be found in colder areas, and of course, darker fur helps to absorb more heat from the sun. So when they're exposed to colder temperatures, they produce black fur to help them absorb more heat. So if you were to take an ice pack and lay it across the back of a cat or across the back of one of these rabbits, then over a period of time, the fur in that area will start to develop to become darker in order to capture more heat from the sunlight. And you notice that in areas of that animal that are going to tend to get colder quicker, such as the nose and the ears. Genotypes are not like blueprints that specify phenotypes. Phenotypes are going to be generally a product of the genotype in combination with its environment. Some genes are going to be linked together. For example, we tend to see light skin, freckles, and red hair that are carried together. So most traits are going to be passed on as independent features. Mendel's law of independent assortment is going to help us find how this is done. So Mendel believed that all genes behaved independently. He did not know that they were on a chromosome. He believed that they were just free-floating entities inside of a cell. So looking here at Mendel's law of independent assortment, we see how that one trait doesn't influence the inheritance of another trait. And so they're going to use here the cleft chin. So with red hair and freckles, these are going to be genes that are going to be found on a chromosome that are sometimes inherited together. So with most redheads, they're going to have, like we said, pale skin, red hair, and freckles. And they seem to travel together with each other. And so this goes against what we knew of Mendel's law of independent assortment. So why do most redheads have pale skin? It's going to be because of these linked genes. And basically they're genes that you find on the same chromosome or maybe even right next to each other on that chromosome. So the same location or loci or locus. So there's going to be about 25,000 genes in the human genome. Yet we have only 23 unique chromosomes, 2 copies of each. Thus, genes influencing some traits, possibly hair color and skin color and pigmentation, must be on the same chromosome. Maybe even right next to each other. So when they are, we say that they are linked genes. They travel together. So looking here at these linked genes, you're going to see these homologous chromosomes. You have 2 for the mother, you have 2 from the father there. If they get close to each other, they can cross over, they can switch sections, and that's when it happens during meiosis 1, specifically prophase 1. Then they go through, they separate, and one of each of the 4 goes into an egg or into a sperm. That's going to end us for this section. We will pick up with our last section for Biology 101 with Unit 8.